Okay, in part 4 here, let's switch gears. In part 2, we talked about the evolution and dominance of gymnosperms. In part 3, we took a break and started talking about physiology and how water is moved under tension up all plants that have vascular systems. Here in part 4, I'd like to talk about undifferentiated tissues, which are called meristems. And plants have a number of different parts and locations in which meristematic tissue persists. This is undifferentiated tissue that allows the plant to grow on an annual basis in different directions, so that we will see that there are meristems to control the production of the vascular system. There are meristems that control the development of shoots, and there are meristems that control the differentiation and growth of root systems. We can get some idea of these systems by looking at a cross-section here. This is a cross-section of a pine stem, and what you can see is the spring growth and the summer growth, with the thicker cell walls in the summer, the thinner cell walls in the spring. So we have 1 year of growth, a second year of growth, a third year of growth, a fourth year of growth, a fifth year of growth. So the oldest tracheids are going to be the ones at the center. Where the plant begins to grow is here in a tissue called the vascular cambium. The vascular cambium is undifferentiated tissue, and in this part of the cambium, it divides to produce new xylem cells, in this case, new tracheids. On this side, the vascular cambium divides to develop new phloem. Well, we haven't talked about it yet. The xylem system is with water under tension. The cells are dead, and they are produced by division associated with the vascular cambium. Tracheids are associated with the transport of water and of nutrients coming up from the root. It's a one-way movement system. On the other hand, once photosynthesis has taken place, we need an export system to move the carbohydrates from the leaves or needles down to other parts of the plant. So the plant has developed essentially an up elevator and a down elevator. And the down elevator are called the phloem. In contrast to the xylem, the phloem cells are alive, and they're driven by positive, movement is driven by positive pressure. So what we have here is the undifferentiated tissue, the cambium, producing phloem, the downward moving elevator, and xylem, the upward moving elevator. In this case, the xylem are all tracheid cells. On this side, they are phloem cells. This indeterminate growth associated with meristems allows tissues to develop, or allows growth to develop in a number of different directions. This means that plants have a mechanism for foraging by allowing growth in different directions in response to resource levels. So the first meristem, undifferentiated tissue, is called the apical meristem. It is called the primary. It is the first tissue that we're going to talk about. It is associated with the production of new leaves, stems, and the decision to go from producing vegetative material to producing reproductive material. This is the apical meristem. These are leaf primordia that are being developed. So in this case, what we have is a vegetative tissue producing a shoot and leaves. Similarly, if we were to look below ground, we'd find root meristems. And root meristems are undifferentiated tissues at the bottom, which are going to elongate, produce new xylem, they produce phloem cells, and they produce root hairs associated with taking up nutrients and water from the surrounding environment. We have another third meristem we call the Cambiomeristem, and it's adopted the name secondary. And this allows for continuous growth, as I mentioned earlier. Here we have the vascular cambium, and on one side the cells divide to produce new xylem, and on the other side they divide to produce new phloem. So we have three different meristematic tissues that allow for indeterminate growth. So unlike animal systems, plant systems are able to develop new shoots, new roots, and new vascular tissue because of these undifferentiated tissue regions. So if this was a clicker question, I would ask you to point to the cambium. Here is the xylem, here's the phloem, here is the cambial region. At this point here, it's useful for us to do some comparisons now because we'll be finishing up our discussion of plants, and it's worthwhile pointing out that both plants and animals are derived systems in terms of the history and evolution of life on Earth, and they have several distinct features, several distinct differences. Plants, like archaea and bacteria, have cell walls. Animals do not have cell walls. They cannot support themselves under tension, and so they would lyse in a hypertonic or hypotonic solution, whereas that cell wall allows a plant to maintain structure under a variety of different hyper and hypotonic solutions. Plants have mitochondria, so do animal systems, but plants also have chloroplasts. Plants have a vascular system, a one-way elevator moving water and nutrients up, and a one-way elevator moving food and other resources down. In contrast, animal systems have a circulatory system, so it's a closed circulation system. Plants have indeterminate growth. The shape is determined over time, whereas animals have a predefined determinate growth pattern. Plants are autotrophic, animal systems are heterotrophic. So the consequence is that plant size and canopy shape are flexible. So that is determined by the activity of the apical meristems, whether it's a shoot apical meristem or a root apical meristem. Whereas growth in animal systems is predetermined. So it's predetermined by cell division growth activities. So this is a brief summary of differences between plants and animals.